Good afternoon, everyone, and welcome to our February Lunchtime Lecture. We're so glad that you could join us today for Bees, Wasps, and Bacteria, Big Discoveries by UT Undergraduates. All right, and I have to say that's the best title we've had in a long time. So get excited. Before we begin, we'd also like to thank Lunchtime Lecture sponsor UT Extended Campus for their generous support. Extended Campus offers graduate programs, professional certificates, and skill building courses to support your career goals, no matter what your next step is. You can learn more about the programs they offer at extendedcampus.utexas.edu. We're also grateful to everyone who chose to donate when registering. Your contributions really help us continue to engage alumni and students across the globe, so thank you very much for that. And on to today's speaker. So, Dr. Joanne Holley, an associate professor of practice with UT's Freshman Research Initiative, where she leads the Bugs in Bugs Undergraduate Research Lab. Her work focuses on the intimate relationships between insects and their symbionts and the ecological and evolutionary factors that shape these relationships. As a member of the Morin Lab, she uses molecular and evolutionary approaches to study these partnerships in natural systems. Dr. Holley also serves as an education fellow with the Office of Excellence, training graduate students and postdocs in effective mentoring for undergraduate researchers. Please join me in welcoming Dr. Joanne Holley.
Hi, everyone. Thank you for the invitation. I'm so happy to be here. I'm happy to talk about the program I work for, the Freshman Research Initiative, and my students and the research that we do. I think it's a fantastic program. Serves a lot of students, and I'm also really proud of my students and everything that they've done. You'll notice I'm Australian. But I've lived in Texas for 11 years, and even though I didn't come to UT, I'm really also very pleased to be part of this community. I really like this university a lot. And hopefully some of that will come through when I talk today. So, as Patricia said, I'm going to talk about bees, wasps, and their bacterial symbionts. So, these are the bacteria that interact with bees and wasps and how they work together to live together. Um, but mostly, I'm going to talk about my undergraduate students and the undergraduate research lab that I run. The insect there, it's a Mexican honey wasp. I'm going to talk about that, but that's going to come in a little while. They're my favorite insects. They're new to Texas. They're from Mexico originally, but they live locally here in Austin and through Texas now. So, what is the Freshman Research Initiative? It's what you'd expect. It's a research program for freshman students. We try and get undergraduates involved in authentic research from their first year at the university. Um, there are a lot of good reasons for why we do this. Um, Part of it is to give them deeper experiences doing research. So they're learning things at depth, not just a lot of things by skimming the surface. But it's also a place where they can get to know each other and a professor and more advanced students in these kind of meaningful, deeper relationships. And so it's part of the College of Natural Sciences, and about 40% of students that join CNS get to join an FRI research stream or a research lab. This year, we have more undergraduates than we ever have. 1,200 have joined FRI, and there are 37 research labs, which is also more than we've ever had in previous years. And so this is just a bit of a taste of some of the research streams. There are four of them. They're all associated with biology or biochemistry in this case, but there's one on robots that's showcased there, autonomous robots. But then there's also uh, an astronomy stream, there's computer science streams as well, physics, but a lot of biology, biochemistry, and chemistry streams because that's where most of the CNS students are focused. And my stream is called Bugs in Bugs because we study the bacterial bugs that live inside other insects. So this is an overview of the program. It started in 2005, and in 2005, all those circles at the top are the numbers of research labs. So there were three original labs with about 50 students, and it grew and grew and grew until about 2014, 2015. That's where it started plateauing, and we got a really big grant to support the program, millions of dollars to build it up. And a big part of that build-up of the FRI was also to have replication sites. So other institutions have copied this model of having research labs, undergraduate research labs. And as I said, there are 37 this year, which is the most we've ever had. And not only are we accommodating more students, but more and more students do want to join FRI. So it hasn't waned in popularity at all. In fact, it's increased. And unfortunately, there are about 700 students on the wait list. And so we try and offer them experiences like this or help them get into research because we can't serve everyone, but we'd like to. And so, of these other replication sites, there's a really big program in the University of Maryland called FIRE, which is based completely on FRI. And there's one at SUNY Binghamton as well, and they serve about 400 to 600 freshmen each year. And then there are programs at University of Texas in El Paso, San Antonio as well. So the program, the model has been really successful, very helpful, and student outcomes are fantastic. They're a bit more likely to graduate, they're more likely to get involved in research, and then really importantly, they're more likely to develop an identity as a scientist. And so my stream, The Bugs and Bugs, started in 2015. So this is our 12th year. And I'm going to talk to you about the way we do FRI. Because there are so many different streams, everyone does it a little bit differently. But one thing that's common for most streams, if not all, is that they are associated with a research lab at the University of Texas. So our principal investigator is Dr. Nancy Moran. Here's a photo of her. She has a research lab that studies the biology of symbiosis, which is how organisms interact with each other. And like us, it's mostly insects and bacteria. And so Nancy's primary research organisms are the pea aphid. This is this pea aphid here with the little babies, honeybees, and then the bacterial associates. We're very interested in finding symbioses that are consistent, long-term, and benefit both partners, the bacteria and the honeybees, but not always. And the other component of the research stream, an FRI research stream, is the research educator, and that is me. So I'm an educator and I teach students how to do research. We don't do the same research as the Moran Lab. We try and fit in with what they do. We might do things that are a little more risky. Um, we might look for bacteria and different kinds of insects. And we have a few of our own systems that are similar to what the Moran Lab does, but different. And so we don't want to we follow in their footsteps somewhat, but we also want to do our own thing. And it's complementary. And so the first 2 years, I'm going to show you the timeline of what FRI is like. And this represents the first 2 years of students' experience in CNS at the university, joining the university in College of Natural Sciences. At their first year at UT in fall, they start by looking at the different FRI streams, visiting them, talking to people from the different streams, and then they can choose which FRI stream they'd like to join. They rank them 1 to 5, and we really try and put them in their first few choices. And about 90% of students get to be in that 1, 2, or 3, which is pretty great. And then in spring semester, they join an FRI stream and then they return in fall. So We started a new cohort just in January. So we're on about week 5 of our new cohort. And then they'll come back in fall to do 2 semesters. And so my stream is set up in a way that we really focus on learning in spring. It's uncomfortable. We really try and push them into the deep end to try and get them to do difficult research pretty early. And so uh, the students are learning just how to be in a lab, how to streak out bacteria, how to use pipettes to move little bits of liquid around, how to isolate DNA. It's kind of difficult because we want them to be as independent as possible. They're also learning the biology of the system, which is quite challenging as well, especially because we learn one thing at depth. So the students can really struggle with that. And they're also learning scientific writing, how to read scientific writing, which is also not very fun. It's not as fun as reading a novel, right? But if they want to be scientists, these are all things that they're going to have to learn. And so we do it in with a lot of repetition and with a supportive environment. So hopefully they're building on this over time. And then in fall, they come back to the lab, and then we're really interested in applying what we've learned in spring. And so the students are on research projects and they're hopefully collecting some data, analyzing data. But this is where I really focus on communicating science. So making sure they can write well, not just read it, but write and also present it. And then we try and tie their experience in FRI to some professional development. So tie it to their career goals. Like, how would you relate what you're doing in the lab to applying for a job? And some students, not all students, but some students also, oh, okay, I forgot to mention this. So having the students for 2 semesters is what we call looping. So the students get to spend 2 semesters with the professor. There are only 35 students each year, so I know them all well. I know what they're interested in. I know what their career goals are, and so do their undergraduate mentors. And so we can try and tailor the way we work with the students for the individual students. And that's where the true mentoring comes into it. And this is really beneficial for the students to get to know each other, to have a sense of identity and a sense of place, but then also to know that they've got people in the ring with them. And so in summer, there's also the opportunity to do research fellowships. I'm going to talk a bit more about that, but this is when the real research happens. Students have 8 dedicated weeks and they are working half-time or full-time conducting research, collecting data, analyzing data. And the students that stay over summer, we do some of the communicating professional development. They essentially serve as role models in the fall. And so students can also stay within the stream for longer and they can stay as a mentor where they're helping the new students learn, or they can be researchers themselves and conduct research. So some students can stay with FRI for 3.5 years, which is nice. It's really nice to have students for so long. You form really deep connections with them, or some students might stay just for another semester or 2 semesters, and that might be enough to give them a bit of a launching off to do something else, like joining another research lab. Oh, there we go. Okay, so this is my students from last night. There's my Monday night session. All the students wearing lab coats are my freshmen. And then in the back, there are some mentors as well. And so these students are hopefully getting to know each other, getting to know the lab space, getting to know the mentors. If they're pre-meds, maybe they're talking to the mentors at the back, Toby and Brett. They just applied to medical school. Toby got into Texas Tech. So maybe they're talking about going to med school with them or what classes they should take. They're probably feeling a little uncomfortable. So gaining confidence and making some friends, that's the goal. And then each week becoming a bit more independent. And so this is my cohort from last year. I had a smaller group, 25 students, and then along the front you have the mentors. So those are current undergraduates that have a bit more experience that are helping them, the newer students out. And a good number of them, about 15 of those students, have stayed on with the stream for this year. And then in the sophomore year, second semester sophomore year, we're really trying to apply what we've learned, as I said earlier. And this is when I'm trying to get the students to do more data analysis and mentoring others. So they learn for the first year and then they can apply that by teaching other students. And in this photo, you can see this is one of my former students, Ethan. He's a mentor and he's showing a student how to use an instrument called a pipette. And then in the third and fourth year, this is where not so many students stay with the stream, but the ones that do are really building leadership skills. They do things like help me with the grading, they run labs, they help with things like purchasing of items in the lab. This is really important for pre-med students. Um, they're communicating as well. They've got to communicate complex ideas. And this is where we get most of our research done. We do want to do research that can be published. We want to contribute new knowledge to the world, and we want the students to be doing that. And so this photo is a picture of my student, Liz Reed. Liz is applying to graduate schools now to do environmental research. And she's presenting at a conference called the Undergraduate Research Forum, which is hosted by CNS. And hundreds of students make posters and then they talk to judges and other students about their research. It's a phenomenal event. It's in April. If you're on campus, you should come. It's really great. It's a fantastic experience for the students because they get to engage with people about their research. They also make a poster, so they're learning how to communicate. And also, they're interacting with each other all together, so they get to see what a vibrant community, research community CNS has for all the undergraduates. And so, put together, the Freshman Research Initiative Undergraduate Research Lab is a sophisticated mentoring environment. So there are two main provisions for mentoring, and the first is career support, so helping your mentees develop so they can have successful careers. And then the second is psychosocial support, where they can get emotional support as well, particularly if they're struggling or even providing it to other people. And so FRI works both those important mentoring functions into the structure because the students are interacting with their peers closely, near peers, which is their mentors. And then on top of that, they know a professor very well, very personally. And because it's a network of 37 different labs with an opportunity to share their experiences, they're connecting with other labs as well, so a broader community. And these are the reasons why the Freshman Research Initiative is so successful and has been copied a lot of other places. Okay. So these are two of my students, Natalia and Aaliyah. We're out collecting insects. And so a couple of weeks ago, I asked the mentors, I was like, what did you get out of this in terms of connections? And repeatedly, they said it was just an easy way to make friends. I could form connections straight away, and that can be really hard. And so by having students all together in the shared space, they can make deep connections. And Haley, she lives across the road from one of her lab mates. Her roommate is dating his roommate, right? These are tight connections that I hope last them through their life. Another really important aspect of the Freshman Research Initiative is we can support students by giving them credit, but also pay. Some students have to work in order to get through their time at university. And if we can give them money, then that means they don't have to work at another place, another shop or something like that, and we can pay them to do research, and then they're building skills that they'll use through their career. Not to say they won't at other places, but they can build some skills in research or mentoring others in research, and that's really, really valuable. We try and do that whenever we can. And so students get paid to mentor. That's built into the FRI system. And then on top of that, the summer fellowships, those are paid through the Freshman Research Initiative. And the Moran Lab, Nancy Moran brings in, NSF grants, USDA grants, and those come with stipends to fund our students. And on top of that, I had a collaboration with the Biodiversity Discovery Stream. We got a 3-year Stengel Wire grant through biology, CNS. And for that program, we could pay 24 students to have summer fellowships or research funding over the semester. And that's hugely beneficial that a student can get paid $5,000 just for a couple of months of research. Very, very important for their progress, but also very important for us and our research progress as well. Okay, I'm getting close to the end of the FRI part, and then I'm going to talk about our research. But this is a photo of us from last fall. We were out at the Hill Country Field Station. It's a gorgeous field station, which is joining the CNS, the College of Natural Sciences Field Station Network. And there we are, we're out collecting insects towards the end of the season. It's a lot of fun. I think that these field experiences can be transformative, perhaps because it was for me, but it's also a great way for students to go out together and do something together and experience together that they might not ordinarily do because they live on campus and they're around campus all the time. So we can drive out and go collect insects. This can be important. I've been mentioning pre-med students a lot, and that's because pre-med students a lot join my stream because it's a biology stream. But about 90% of all biology and biochemistry students are pre-med when they're in their freshman year. And so this is a chance for them to do something a little bit different and go out and do something else that they might not ordinarily do. So we try and go out and collect insects and have some field experiences. Okay, so I'm going to take a breath because I'm going to change gears. And now I'm going to talk about the kind of research that we do. And I'm just going to show you a couple of our projects. And I want to highlight some of my students that have had contributions and we've published research. So these are 6 of my former students, Lexi, Jen, Anna, Sarah, Nikki, and Tasneem. And we've all worked on publications together. And one of the beauties of having a relationship with a research lab, the Moran Lab, and also the Barrick Lab, is we can work with other graduate students and postdocs, so other researchers. And this is Kate Elston, she was in the Barrick Lab. Layla Phillips, who's a current PhD candidate in the Moran Lab, and Alan Silva Cerqueira. He was a postdoc in the Moran Lab as well. And so they've taken some of the research that my students have conducted and incorporated it into their own. And that's why we have these associations with the PI lab. It's really hard for undergraduates to do full research, but we can contribute to these broader projects, and that's really beneficial. Okay, so insects and science. So we study insects for a number of reasons. One, I have a PhD in entomology. That's my study system. I've been working on social insects since I was 19. But insects are so important. There are over 1 million described species of insects. So that means for undergraduate research, there is so much that the students can learn that they can work on that's new. And insects are integral parts of ecosystems, and they provide a number of free services that help our ecosystems run. I've included a couple of photos here. There is put on the laser pointer. Okay, these are fire ants. This is taken at the Brackenridge Field Station. Fire ants are from South America. They're an introduced species. I'm sure if you're Texans, you know fire ants. And this is a head decapitating fly that serves as biocontrol that can kill the fire ants. And those were introduced by UT Texas as from UT researchers. And then here are some dung beetles. Could you imagine a world without dung beetles? How gross would that be, right? Um, Dung beetles perform a really important service, and so do other insects like flies. They decompose living matter. They can filter water, and then they're also important for recreation. If you like fishing, what do fish eat? Fish eat a lot of insects. And so they provide these important services, and we really need to understand how they live, how they contribute to the world. And if we're going to understand that, we have to understand their symbioses now, so how they interact with their bacteria. And then the one ecosystem service that we really focus on is pollination. So we study pollinators. Again, a lot of pollinators, a lot of pollinators are social insects, which is my study system. And here's a picture of a bee that's covered in pollen. Pollination services in the US are worth about $34 billion per year. And pollination services aren't just honeybees. Oh, honeybees are very important, but there are also a lot of native pollinators as well. And some of them are in decline as our world changes, particularly with climate change. Insects are changing a lot too. And so we want to understand what's happening and track those changes. And pollinations and pollinators are one thing that's changing a lot. That being said, honeybees, even though they're not native pollinators, are a really important study system, and the Moran Lab focuses on them and has contributed a lot to understanding honeybees and their bacteria, their gut bacteria. And so here's a picture uh, of a frame, and you see there's capped pupae in there, so growing pupae and honey as well. Honeybees are really important for pollinating plants, but we also get honey from them as well. And what's great about honeybees is they're domesticated, so we can study them easily, very comfortably. We can keep them. You can order them from the internet and they'll arrive in your lab, and then you've got your study system. It's a great system. And you can have up to 60,000 individuals. And so they feed on two things. They feed on nectar, which they turn into honey. So they feed on honey, and then they feed on pollen. So they have a pretty simple diet. Um, they're small, there's a lot of them, and so that's easy to study. And so to compare the honeybees to humans, humans, you have your own gut bacteria, you have your own microbiome, and it's in a very large digestive system, which is probably about 25 feet long. There are hundreds of bacteria. It's hard to understand how they all interact with each other. And it's also really hard to experiment on. And what you eat is probably pretty different from what other participants eat. And then what we eat in Texas is pretty different to what people from other parts of the world eat. So humans are a very important system, but they're not always easy to work on. Honeybees are much simplified. So if we want to understand how these symbionts, these microorganisms, are interacting with a host, for their own mutual benefit, honeybees are a great system. They have 5 species of bacteria that are present in every honeybee in every part of the world. If you collect a bee from a flower outside, it'll have the same bacteria as a bee collected in Russia, right? But then they have some other bacteria present as well. The other great thing about it is you can grow them all in an incubator. You can put them on an agar plate and they'll grow in an incubator. And so you can do great experiments with them. You can also get bees from a hive and you can clear out all their bacteria and then you can feed them individual bacteria. So it's a really manipulatable system. And on top of that, they only eat nectar and pollen, like I mentioned. So it's a really great system. And this is what the Moran Lab studies. So we don't study that. We take what they know about this really cool system, and then we try and uh, apply that to what we know, apply that to other insects. And so honey feeding social bees. Okay, so these are honeybees. They all have 5 bacteria, right? Bumblebees, they also, even though they're pretty different, they're very cute. They're very adorable. They live in smaller colonies, but they also have these 5 bacteria. And then these are stingless bees. This is a photo taken of a stingless bee hive in Colombia. You know what? They have these 5 bacteria. Some stingless bees have lost 2 of the bacteria and probably acquired new species, but they also are really similar. And so, knowing what we know, my students and I are like, well, what about other insects? What about honey wasps? So, this is a honey wasp. Honey wasps have big paper nests. This is a nest, and you know the wasps that live outside on the eaves of your house. They're related to them, and they rather than have the nest that's shaped like an umbrella, they're just round because they just keep adding to it and adding to it. And so this might look a little familiar. These are pupae, so these are wasps that are pupating. And then you have all these little cells, and in the cells, there's either eggs and larvae, or there's honey. So honey wasp nests can get up to 20,000 individuals. Honey bee nests can get up to 60,000 individuals. So they're pretty big. And the other thing is they to start a new nest of honey wasps, they split in two. So you always have constant interaction among individuals, and you also you're starting a nest with a group of workers. So that means there's always a mode of transmission for bacteria to be shared among different individuals. And so this is a honey wasp. I think they're the cutest, the prettiest wasps there are. You can tell it's a paper wasp because of its wings, they're folded. But what's weird about the honey wasps, and most wasps aren't like this, all wasps, they can only sting once. They have a barbed sting that'll stay in your skin, which is like honeybees. So they're a lot like honeybees in a lot of ways. So we wanted to know what kinds of gut bacteria they had. Are they like the honeybees and the bumblebees? And so this is in 2015, just after I got here, I reached out and got hold of this person. This is Gary Rankin. He's a beekeeper from south of San Antonio, and he loves honey wasps, and he would bring them onto his property. And here is a huge honey wasp nest, and the black things all over it are honey wasps. And that nest is about 3 feet long. And you can see this is the paper structure because they're a proper paper wasp. And so my students, Lexi, Jen, and Anna, we worked on this project together. Lexi and Anna did a lot of the data analysis, and Jen did some of the artistic work for the project. And so this was over several years, and a few other students have contributed to this as well, but these are the students that were on the publication. So we collected, this is the range of honey wasps just showing from Panama up to Texas. They go up to Waco. What's cool about honey wasps is they were only common along the border up until maybe 30 years ago, and they've moved north, presumably as Texas has got hotter. And they're also found over kind of into the border of Arizona. And there are 15 species of honey wasps, but only three are known to store honey. And so honey wasps are new to this part of Texas. And so that's kind of interesting itself. But we collected honey wasps from Austin down to the border. So from all these different sites. And then we dissected them. This is the crop, the social stomach, the midgut, the hindgut, and that's the venom sac, and that's the sting. And so we took this gut section and then we extracted the DNA and then we used computational methods, so data to work out what bacteria were present. And so we did this in honey wasps, but we also looked at some other wasps as well, because if we find something in honey wasps, maybe other wasps have the same thing. And then it's not specific to honey wasps, is it? So these are Polistes paper wasps. These are the ones that live in those kind of umbrella-shaped nests outside your eaves. And so we looked at four different species of Polistes. These are social wasps. And then we looked at seven different species of totally solitary wasps. So wasps that don't form colonies at all. And then this next slide is kind of intense, but I do want to show you some data because it took a long time to generate and my students worked hard on it. So what we're going to see is a big panel with lots of different colors. And then this section up here is going to have lots and lots of lines with different colors. And each color represents the presence of a particular species of bacteria. And so these are the honey wasps and these are the other wasps. Okay, alrighty, a lot of data. And that's what it looks like. So the takeaway here is we're looking for a general trend. And these are the wasp nests that go from Austin all the way down to Harlingen along the border. And every one of those wasps has this blue bacteria and this purple bacteria. And this blue bacteria is a lactobacillus, and the purple bacteria is a bifidobacterium. So they all have bifidobacterium and lactobacillus, kind of like yogurt. Yogurt's like that too. And so what this tells us is that the honey wasps have a conserved microbiome. And then these are the other wasps, and their microbial communities are much more diverse. Some are small, some are big. They're not as consistent. And so that tells us that honey wasps, they're doing something special. And that even though these social wasps interact with each other and can share their microbial communities, they don't as much. They're not so conserved. They might share them a bit. And then this is a heat map. And every time you see a light rectangle, that shows you that there's bacteria. And if it's very bright, there's a lot of bacteria. And this whole group here of these different bacteria are all pretty much only associated with honey wasps. So they have their own unique microbiomes. So Jen did this beautiful artwork for this project where we looked at where the different bacteria associated with honey wasps come from. And it turns out that bacteria comes from bumblebees and honeybees and also other wasps as well. So they share the same bacteria that honey feeding bees also have, and then also some of the wasps as well. And they're not getting it, they're getting some of it from the flowers, but not very much. So this tells us that they have a conserved microbiome, and we think they've had it for a long time. And we think it's probably these bacteria are probably doing something to help the hosts, but we don't know that yet. Okay. And so the takeaway from this is that honeybees, bumblebees, and honey wasps all have conserved microbial communities. They all have a microbiome that's shared among individuals. But paper wasps, they don't. These other paper wasps. And we think it's because of their diet. We think the switch to feeding on honey allowed honey wasps to get these new bacteria, different bacteria. And honey wasps, they feed on nectar. And then this paper wasp, in its mouth, it has chewed up, chewed up caterpillar. We think that it doesn't have the microbes because it doesn't feed on honey. Okay, so that's one of our main projects. It's still ongoing. These are two of my students that work with me in the Moran lab. So they're paid off an NSF grant. This is Elizabeth and Zoe. And we've isolated some of these bacteria. One of them's called Bifidobacterium texanum. We're going to name it after Texas. And then this one down the bottom is the thing called Schmidhempelia, which was only known from bumblebees, and no one's ever grown it in the lab before. And so we're trying to work out what sugars they digest. Because if they live in the gut of a sugar-feeding wasp, then we think they're probably able to break down sugars. And then these are some of my students, Sarah, Alex, and Raj, and they're helping describe this species officially so then it can have a name. Okay, I'm going to start maybe a little faster. Okay. And another project that we worked on was carpenter bees. You might know carpenter bees because they can drill into the wood of your house. So my student Nikki is like, I want to work on carpenter bees because they're really cute. I was like, they are really cute and they're abundant locally. So Nikki collected some carpenter bees, and Anna went and collected a ton more, and we looked at what bacteria they had. And here's a photo of one of the local ones, Xylocopa virginica. I'm sure you've seen it. If you live around Austin, it'll be on your flowers. And so we looked at 4 species of carpenter bees, and then we looked at 5 species of solitary bees. And you know what we found? The same thing. In this case, the social bees, they have a conserved microbiome. And those bacteria are a lot like the honeybee bacteria and the bumblebee bacteria too. But these solitary bees, they don't have them as well. And so by being part of a social colony, they can share these bacteria with each other. And they have a very different lifestyle. Carpenter bees, they live in wood. See, these are the panels where they live, and these are channels. You can see there's a line drawn. And what a carpenter bee does is lay eggs in all of these chambers and gives them some pollen mixed with nectar, and then the eggs develop. But here's an example. But the egg-laying females will share spaces with other females because it's really hard work to dig out chambers like that. And that's enough for them to share bacteria, which is probably enough for them to maintain these full microbiomes. And so back to the students, what are the implications of doing this kind of research? Well, we've got 2 publications. These are published in peer-reviewed journals, one in Applied and Environmental Microbiology and the other in MBio, that's the name here, MBio. And these are journals that are associated with the American Society for Microbiology. They're excellent journals and they're reviewed by other scientists, and the students have created new knowledge. This carpenter bee paper was published in not 4 years ago and it's been cited 40 times, and this came out just last year. And so students contributing new research has a really great impact on their future success for getting other positions, particularly going into graduate school. if they want to continue on in science, that's a true indicator that the student can be productive, can conduct research. But it's also great for other fields, especially pre-med, because again, it shows that the student can apply themselves and do difficult work over time. See it all through. And the other thing is now my students with this training, especially after a couple of years, they become experts themselves. And so Sarah, my former student, she's works in a bacteriology lab at a hospital in Houston now, but she collaborated with Alan to describe this bacterium that was isolated from the carpenter bees. We're calling it Nephella. It's named after a local bee specialist. And Tasneem isolated this bacterium. It's called Orbis moriae from a fly. And Tasneem contributed to a publication led by Kate and Layla. And then Layla described this species and they collaborated together to choose a name. And Tasneem got to choose the name for this bacterium and she named it after Ruth Ella Moore, who was the first Black woman to get a PhD in natural sciences. And then now my students are collaborating with other streams. This is Haley, and Haley's been working on a data set from the bioprospecting stream. She's able to do this data analysis where we looked at the microbial communities, the bacteria associated with superworms. Some of those bacteria can break down plastics, and that's what the bioprospecting stream does. And now my students are working with some students from the fish behavior lab to teach them how to do DNA extraction so we can look at the microbial communities, the microbiomes of fish as well. And so they get to contribute to these larger projects, but they get to come in with some expertise, which is a really phenomenal thing for someone so young and still in training to be able to contribute. And they also learn these transferable skills. So they're learning how to do research, uh, and they're learning how to be in lab, but they're also learning these core competencies that can help them with other jobs, communicating, working in teams, troubleshooting. And each of these students has continued on. Nikki went and got a master's looking at vector-borne diseases, diseases vectored by insects. But I included Leo here because Leo went and got went to UCLA to study data analytics, and he presented his research on the microbiomes of dung beetles, and it was really surprising to them, and it had a big impact. But a lot of my students now are doctors or med school, and this is Dylan. He's one of my first four students in 2015, and he's at Baylor College of Medicine, and he's in his last year of residence. Um, he's he was a goofball, he's still a goofball, and I hope that his time with FRI and with our stream left a mark on him, and here he is dressed as a moth for Halloween. I just took this from Instagram. And then Lexi and Jen, I highlight them. Here's a photo of Lexi leading a session, a discussion section during the pandemic, but they're both doing PhDs in entomology and they both study bee microbial communities. Lexi's at UC Davis, Jen's at UC Irvine, at fantastic colleges, the best there are, and they're going to have continue to have big contributions. They have really bright futures. I'm so proud of them. And so FRI, the Freshman Research Initiative, and my lab can not only produce valid research, but we can have huge impacts on these students. And I've had 300 students come through the lab over the last 12 years, and they're doing really great things. And so with that, I'd like to finish up and say thank you for your time. And if you have any questions, I'd be happy to take them.
Thank you so much. I have a question. I'll just start us off.
Yeah.
So this is kind of a silly question, but I'm very curious. What does wasp honey taste like?
It tastes, good question. I get asked that a lot. So wasp honey is like honey, bee honey, because they're collecting nectar from the same sources. However, they store it in paper, and I think it tastes bitter. To
Because it's
me,
paper. Yeah.
because it's in paper. I think it tastes bitter. Not everyone can taste that. It's like it picks up some of the tannins, but it's like honeybee honey in that the nectar is going to dictate what it tastes like. It's more runny. They don't dehydrate it, concentrate it as much.
Okay.
Yeah.
We do have our first question in the QA, and it's, is there an application for this research?
Yeah, potentially. So one thing, so in terms of the honeybee research, you can go to the store and you can buy probiotics for yourself that can help keep you healthy. We've been interested in doing that with the honeybees as well because they got these five bacteria. Maybe um, you can feed the bees these bacteria and that'll keep them healthier. Bees get stressed. They get very stressed with drought, pathogens, also some pesticides, or so if we can give them probiotics, it can keep them healthier. Because one thing we do know about these microbial communities is they're kind of necessary for reducing some of that stress, protecting them from pathogens and keeping them a bit healthier.
Got it. Is there a link between the bacteria and the quality of the honey?
I don't know. I don't
Uh,
think so.
okay.
Uh, I don't know. I don't think so. So the honey is produced because the bees feed on it, and then everyone knows this, I think they spit it out, they regurgitate it, and then they suck it back up and they regurgitate it again.
I
And
gotcha.
so it's in their crop, which is their social stomach, and then it's kind of in the nest. The bacteria live in the hindgut, in
Okay.
the so I don't think the bacteria are interacting with the honey that much. That's my
Okay.
suggestion. Maybe, I
Okay.
don't know.
It's a good one. Here's another question. How do you identify which bacteria are present? Is it a microwave detection?
We do it in two ways. So the first thing we do is we extract all the DNA from the guts and then we sequence it. So we send it off to a lab and then they send us back a document with lots and lots and lots of DNA, like strings of DNA. And then we use computational methods to work out what all those strands of DNA are. And then we can estimate what the bacterial community is based on the DNA. So that's the first thing we do. That's kind of challenging. And I showed you a picture. That's what my students do. That's kind of hard, and that's what makes them experts. So we work out what bacteria are there, and then we can be very targeted and try and isolate the bacteria. So we grow it in the lab. Um, these bacteria don't like to live outside of their bees or their wasps, and so they don't like to grow in a lab. So we give them super rich media, and then we grow them under CO2 conditions, and sometimes they grow, sometimes they don't grow. So two-pronged approach to get the bacteria.
Got you. Has anybody checked varroa mites? I think I'm saying that correctly for the bacteria.
Uh, I don't know. I don't know. I'm not sure. Um, so varroa mites are associated with honeybees. They're a pathogen. They were in Apis cerana, not Apis mellifera, and jumped over. So they're quite a problem. Um, so the bacteria are associated with the honeybees, so I don't think they'd be associated with the varroa. But I can tell you a cool thing that the Moran lab developed with the Barrick lab, and that was they used one of the bacteria. It's a symbiont called Snodgrassella. They could engineer it, and it could kill off, reduce the varroa mite, which was really cool. So they could use it to decrease varroa mite presence and also a couple of other pathogens as well. And so that's one of the cool things about this honeybee system is the bacteria can be engineered to do other things.
One other question about the honey. Have you looked at the bacteria in the honey produced by the various bees and wasps, and what have you found?
We have not.
Okay.
And part of the reason is but, honey is super highly concentrated in sugars, um, and there's very little water in there. So I'm not sure how much bacteria really grows in there because it's a preservative, right? It's preserved and it has other enzymes in there as well. So I don't know. I haven't really looked at the honey, in part because we really want to know what bacteria are associated with the host. And the honey is kind of transitional through the host. And we want to know if the bacteria are forming biofilms, like layers within the gut lining. And so that's where we focus rather than on the honey so much.
Okay.
That's cool. Like, it's a cool question.
Yeah. Um, all right. So how has funding been impacted over the last few years, specifically in regards to what appears to be a challenging environment these days?
We're only a year into the change in funding, the funding landscape. So we haven't been impacted.
Okay.
Yeah, hope hope it stays strong. That's certainly what we want. But one thing is the Freshman Research Initiative is funded a lot by donors, thankfully. So by instructional funding, but a lot of former UT people actually provide funding to support this, in part, sometimes for labs that they feel, an affinity for, or sometimes for the whole program. And so actually most of those scholarships, that money comes from donors.
That's beautiful.
It really is.
With more students applying these days, are there spaces for them, and how are students chosen to be in the programs?
Good question. So there aren't enough spaces. It's a real shame. That's why we have these wait lists. But we do try and accommodate as many students as we can. We think it's so important. So the students come from different pools. Some of the students are honors students. A lot of the honors programs offer a place in FRI. And then students can apply themselves. And then there are other programs like TIPS, the TIPS scholars. Gosh, I wish I could remember what TIP stands for. Uh, Oh, I can't remember. Sorry. I'll have to look it up. So the tip scholars, all of those students get a place in FRI as well. So that's around 250 students each year. And then other programs as well. They get to come straight into FRI and then students can apply themselves. They get invited to apply.
Okay. One last question, and then we're going to have to close things down. Does artificial intelligence play a role in all of the data analysis that your students do, that your teams do?
So some streams use it more than others. I think the autonomous robots, particularly, and then we have some of the computer science ones. In terms of the data analysis we do, there's one section of our data filtering where we do use models, iterative models, which you would incorporate, which would kind of come under the umbrella of artificial intelligence. And we do that to provide a model for sequencing errors for the instrument that sequences the DNA. So we do use a little bit of AI. I think most people do. And then we can incorporate some AI models into our teaching, actually, which can be cool. You can train an AI model to help answer student questions. That would be another application. So we think about it and use it more
Yeah.
as it becomes available.
So Tony let us know what TIP stands for, Texas Interdisciplinarian Plan. So
Thank you. Thank you.
no, thank you, Dr. Holley. We really appreciate it. And thanks to everyone for joining us today. And this was fascinating. We really appreciate your time. And thank you again. Our lunchtime lecture series will continue next month. And we'll be sharing more information soon at texasexes.org/lunchtime lectures. Y'all have a wonderful week and thank you.
Thank you.
Thank you.